Hello dear students. This is our third lecture on the topic enzymes. In previous two lectures, we have already studied about various aspects of enzymes. In first lecture, we studied first the general properties of enzymes. We studied what are the components of enzymes, that is protein part, non-protein part, organic part, inorganic part. Then we also understood the mechanism of enzyme action, that is how the enzymes lower the activation energy. We also studied the mechanism of lock and key hypothesis, also induced fit hypothesis. And in second lecture, we studied various factors which are influencing the rate of enzyme action, which may be the temperature, pH, then the enzyme substrate concentration, as well as different types of inhibitors. In today's lecture, we will cover two very important aspects of enzyme study, which are nomenclature and classification. Just like the different rules which we follow for naming all biological organisms, like be it plant or animal, bacteria, virus, likewise, there are certain rules which are framed by International Union of Biochemistry and Molecular Biology for naming and classifying the enzymes. So let us study them. First is the nomenclature. Nomenclature is the science of naming the individual. So just like we follow ICBN, ICZN, ICVN, all these you have already studied International Code for Botanical Nomenclature, Geological Nomenclature, Virological Nomenclature, and so on. Likewise, here also we have certain rules for naming enzymes. So when we talk about nomenclature of enzymes, there are three parts in naming any enzymes. What are these three parts? The first part is whenever we name any enzyme, the name is with respect to the substrate on which that enzyme is active. So the first part of the nomenclature is the name of substrate. Then the second part of the nomenclature is the type of reaction that enzyme is guiding, that is catalyzing. Okay, so the second is the type of reaction. And the third part of nomenclature is the suffix we use, the end part of the enzyme. Suffix ase, ASE, is given to any name of the enzyme. So let us understand these three parts, that is name of the substrate, name of the reaction, and the suffix ase, by taking one example. For example, the enzyme lactate dehydrogenase. When we talk about this enzyme, so there are three parts. One is the lactate part, second is the dehydrogen part, and third is the ase part. This means the enzyme lactate dehydrogenase acts on the substrate lactate, that is lactic acid. Then it catalyzes the dehydrogen type of reaction, means it is basically acting as a redox reaction. Okay, it is removing the hydrogen. Okay, it is reducing the substance. Okay, or adding a hydrogen, adding a hydrogen, or it is removing the hydrogen. Okay, so that is redox reaction. So redox reaction, always they are coupled. One thing, if it is reduced, other will be oxidized. So they are coupled together. So the type of reaction it is catalyzing is redox reaction. And then third is, here ase is the suffix we use. So if we call an enzyme lactate dehydrogenase, it follows these three parts of nomenclature, that is we are including the name of the substrate, we are also including the type of reaction it is catalyzing, as well as we are using the suffix "ase." So this is the way how we name the enzymes. But there are few examples also, because before these rules were formulated, certain enzymes were already named, and at that time, in previous times, the name which was used, there the suffix used was "in." So for example, when we say pepsin, so here suffix use is "in." Likewise trypsin. Okay. So this here, because they were popularly used quite before these rules were framed, therefore we still use them, like pepsin, trypsin, chymotrypsin, like this. Also, there are certain enzymes where we don't specify the chemical reactions they are catalyzing. For example, maltase, lipase, sucrase, all these things. Otherwise, these three parts must be included in the nomenclature of the enzyme. So this was the first part, that is nomenclature. Okay. So now we will switch over to the next part, that is classification of the enzyme. Now let's switch over to the classification of enzymes. As per International Union for Biochemistry and Molecular Biology, the enzyme classification, enzymes are classified into 6 classes, and these 6 classes are in this order. First class is called oxidoreductases. Class 2, second class is called transferases. Third class, class 3 is called hydrolases. Class 4 is called lyases. Class 5 is called isomerases. And class 6 is called ligases. So mnemonics you can write OTHLIL. So these 6 classes, they catalyze specific types of chemical reactions. Let's go into the details of these chemical reactions. You will understand it in better way. So the class 1, first class is called oxidoreductases. Those enzymes which catalyze the redox reactions are called oxidoreductases. For example, if there are two substrates, substrate A is in oxidized state and substrate B is in reduced state, then this enzyme oxidoreductase will catalyze in such a way that A becomes reduced state and B becomes into oxidized state. So this is the first class which catalyzes the redox reactions. Let's take some example. Here, lactic acid, that is lactate, which is in reduced state, whereas NAD, nicotinamide adenine dinucleotide, it is in oxidized state. In presence of the enzyme lactate dehydrogenase, whose nomenclature we studied, what will it do? It will convert lactate into pyruvic acid, which is an oxidized form, and NAD is reduced to NADH2. So this is how this enzyme oxidoreductases, they convert they catalyze the reactions. Commonly, they are also called as dehydrogenases, reductases, oxidases. Wherever this term comes, all these enzymes belong to class 1, that is oxidoreductases. This is class 1 of the enzyme classification. Now, class 2. Second class of reaction are transferases. As the name suggests, they transfer something. What they transfer? They transfer one functional group from one place to other. For example, suppose in this reaction, there is a substrate A. To this substrate, this X is one functional group. So, this enzyme transferases will catalyze in such a way that this functional group X is transferred to another substrate. So what will become? So this from this A, the X functional group will get removed and it will get attached to the next substrate BX. Okay, so this is how this transferases, they catalyze the reactions. Let us see some example. For example, glucose. To glucose, this is ATP. ATP has got 3 phosphoric acids, that is adenosine triphosphate. So, the enzyme, that is hexokinase, enzyme hexokinase, what will it do? It will transfer one phosphate group from ATP to glucose. So, what will become? So, the product will be glucose 6-phosphate and this ATP will get converted to ADP, adenosine triphosphate, which means one phosphate group has been removed from ATP and that phosphate group is attached to glucose. So this is the product. Okay. The second class is transferases. They transfer some functional group from one substrate to other. Now comes class 3. Class 3 are called hydrolases. As the name suggests, hydrolases means they break any substance with the help of water. So, they are basically breaking big molecules into small in presence of water. For example, if we see here, this is one substrate. When it reacts with water, it will break up into two parts, hydrogen parts and hydroxyl part. So, hydrogen part will get attached to this part and to this X part, OH part will be attached. So, this molecule, which is a bigger molecule, gets broken down into smaller molecule. So, this, as a result, these two will be the products. For example, when maltose is a disaccharide which is made up of 2 glucose units. Okay, and these 2 glucose units are held together by glycosidic bonds. So when water comes, reacts in presence of the enzyme maltase. So what will happen? This maltose will be broken. As a result, this of water, there will be 2 glucose molecules produced. The glycosidic bond will be broken. So big molecules are broken down into smaller molecules by this enzyme only in presence of water. If water is not present, this enzyme will not work. So this was about class 3 hydrolases. Next comes class 4, that is lyses. These enzymes also break bigger molecule into smaller, but here water is not required. So because water is not required, so it will break up by forming double bonds. So let us see. Here, this is one compound, and when this enzyme, here we will write H stands for hydrolysis and here lysis. When this enzyme lysis, it acts on this big molecule, it will break the molecule into 2 parts. So as a result, this A and B, they will share the bonds. So this single bond has converted to double bond and these two will form the bond. So as a result, the product is formed. Okay. So basically, they are decarboxylase. So one carbon dioxide will be removed. As a result, there will be double bonds formed. For example, when fumarate, that is fumaric acid, it reacts in presence of fumarase. What happens? Carbon dioxide is removed and this will result into the formation of malate. Okay, so class 4 is ligases. Lyases, sorry. So class 4 is lyases. And now class 5 is isomerases. As the name suggests, isomers. What are isomers? Isomers are those chemicals which have the same molecular formula but different structural formula. So here also, these isomerases will form isomers of some constituents. So they will convert the compound into either cis form or trans form. Okay, so for example, this is there, this one substrate is there, that is converted to its isomer form, that is A to A dash. Then that enzyme will be isomerase. Okay, for example, glucose 6-phosphate is converted to fructose 6-phosphate. Molecular formula of glucose 6-phosphate and fructose 6-phosphate is same. Example, hexose isomerase. So this enzyme will convert the glucose 6-phosphate into fructose 6-phosphate, which is isomer of glucose 6-phosphate. So class 5 is isomerases. And class 6 is ligases. Ligases are also called as synthetases. As the name suggests, they are synthesizing big molecules. For example, here A is a small molecule, B is again a small molecule. So in presence of this enzyme ligases, so these two A and B will be held together and they will form a big molecule. For example, this is ADP, adenosine diphosphate. When it reacts with inorganic phosphate in presence of an enzyme ATP synthetase, a big molecule is formed, that is ATP. So this is called synthetase. So these are the 6 classes of enzymes catalyzing a specific type of reaction, which are basically the basis for biology. I hope you have understood. You must remember this mnemonic OTHLIL. Again, you should know O stands for oxidoreductases, T stands for transferases, H stands for hydrolases, L stands for lyases, I stands for isomerases, and finally L stands for ligases. So these 6 types of enzymes, they catalyze specific types of reactions. Okay, so today we have completed this nomenclature as well as the classification of enzyme action. These are very important, not only from exam point of view, but they will be used at various places in physiology, be it plant physiology or animal physiology, human physiology, or in biochemistry, in genetics. So please go through them. You must be aware with all these reactions and the enzymes. And all these enzymes are reversible. So these enzymes can act on both the ways depending upon the conditions which are prevailing. Okay, I hope you have understood all the aspects of enzymes. Thank you very much.